Hello, everybody, and good morning, and welcome to today's Precisionary Instruments webinar as part of our scientific series. I'm Abby, your Chief Scientific Officer, and I'll be hosting again for this session. So it's my pleasure to introduce Dr. Taran Gujral, who is an Associate Professor of Human Biology at Fred Hutchinson Cancer Center, and he's the Director of the Tracer Rare Cancer Initiative. Dr. Gujral's lab brings together pharmacology, machine learning, and cell biology to uncover new treatment strategies for rare and therapy-resistant cancers. His work includes the development of innovative platforms and tools that use 3D tumor slice models and AI to predict personalized drug responses. And today, Professor Gujral will share how organotypic tumor slices can be used to study cancer stroma interactions and identify new therapeutic targets. So we're very excited to hear more about this powerful platform and its potential to transform cancer research. And with that, I'd like to turn it over to you, Professor Gujral.
All right, thank you so much. Thank you for also this invitation and also a nice introduction. So good morning, everyone. What I plan to do today is present our work on how we are using or the application of organotypic tumor slices in our work, trying to understand complex cell-to-cell interactions between cancer cells and other cell types in the stroma. This work was developed and led by several people in our lab, particularly a former postdoc in the lab, now Nishida Aoki, and also current research scientist Song-Mi Zhu, also with some help from Ramya Sivakumar, also a former postdoc from the lab. So as cancer biologists, we've learned to now appreciate that for most solid tumors, they contain not just cancer cells, but also many other cell types in the stroma. That includes fibroblasts, immune cells, endothelial cells, and all of these other cell types that secrete many growth factors in cytokines, chemokines, and also extracellular matrix that together form this milieu of extracellular of the tumor microenvironment. And so there's a constant interaction between cancer cells and many other cell types in the microenvironment. And these interactions are important, not just affecting how the tumor is going to grow or spread or resist to therapy, we're also responding to various kinds of treatment. And unfortunately, many of our model systems to study a tumor or to study cancers and cancer cells effect, particularly when it comes to a drug response, they lack this complex microenvironment. We do a great job in treating or killing a lot of time cancer cells on a petri dish. But when it comes to treating a patient or making the same drugs effective in more complex models, in vivo models, or animal models, or in patient, the attrition rate is pretty high. And that is because of this complex microenvironment that affects how a tumor in its native microenvironment will respond to a particular therapy. So what are the different model systems that we have developed for the past 75 years as a cancer biologist? So looking at the schematic on the left end of the spectrum are the cell culture models, which are 2D models. We isolate cell lines from a tumor, we grow them on petri dishes, and it gives us a great perturbation capability, which means we can perturb these cells with chemical and genetic means. So we can do genome-wide RNAi, CRISPR screen. We can do thousands or tens of thousands of compound screening, but also gives us the lowest physiological relevance because it's just cancer cells growing on plastic. On the other end of the spectrum are human-derived or model organisms, such as genetically engineered mouse models, which give us more physiological relevance, but it gives us less accessibility for doing a large-scale screening. And one of the model systems that sort of sits in the sweet spot is a tissue slice system. Now, this is a model system that was initially developed in 1960s in the field of neuroscience, where they had figured how to make tissue slices from hippocampal brain, from brain. And these slices were shown to represent the complexity of the intact brain. And these slices were maintaining the tissue architecture. And you could generate a large number of slices for many biochemical and pharmacological studies. So in our lab, we did not invent this method. We adopted very similar approaches that were developed more than half a century ago from neuroscience. And instead, we use a tumor tissue to make organotypic tumor slices. So the setup is pretty straightforward. We make these biopsy punches, about 6 millimeter core, and these are then using vibrotome or compressed-tome are used to make thin slices. The slices can range from 200 micron to 400 micron, and the slices are then placed on this liquid air interface. They are grown on, or they're placed on this insert that sits in a well. So the slices never touch plastic. Slices are never exposed to any enzymes. These slices contain multiple cell types, as shown by images on the right. Using fluorescently conjugated antibodies, we're able to detect many different cell types that are present in slices with half an hour to 1 hour of live cell incubation with the antibodies. Showing here is endothelial cells, T cells. So in a very simplified way, we can see where various different cell types are in these organotypic tumor slices. So if you're thinking about using these slices for pharmacological studies, we also want to be able to develop methods so we can deliver small molecules in tissue slices. And one of the compounds that we use for optimizing this method is doxorubicin. Now, doxorubicin is a chemotherapy that is approved for breast cancer and also many other solid tumors. The advantage of using doxorubicin is that it is a fluorescent molecule. So we can see under the microscope where does it go or which cell types uptake doxorubicin. So using doxorubicin as a tool, we're able to optimize delivery of small molecules in tissue slices. So with some gentle flow of the tissue slices based on these insert, we are able to deliver doxorubicin to more than 90% of cells in the tissue slices. And as we go into this area of generating tissue slices from intact tumor, we also wanted to understand this tissue, the small piece of tissue slice that we're generating, it is representative of the tumor microenvironment. So this work was led by Dr. Ramya Sivakumar, who's a former postdoc in the lab, where we characterize how the different cells in the immune microenvironment represent the entire tumor, and also how they change over time. So showing on the left are the bar plots of PY8119 tumor, which is a breast mouse triple negative breast cancer. So we're using a PY8109. We inject these cells in mice in memory fat pad where they form tumors, and those tumors are then taken and we look at the chunk of a tumor, and then we look at the slice. And we look at the total number of cells in the slice and compare it to the core, and also the total number of immune cells. We also look at various fractions of different types of immune cells using multicolor flow cytometry. As you can see, the slices very well represent. So this is day 0. This is day when we make the slices from the tumor. On the day that we make the slices, they do capture the complexity and also the distribution of various different cell types that are present in the big chunk of a tumor or a core compared to slices that are made from the same core. And on the right are images of many different mouse models that we had used a very similar setup to characterize how well these slices represent the microenvironment. In the breast cancer, pancreas, colon, in each of these cases, we were using mouse cell lines so that we could inject them back into immune competent mice to be able to characterize the microenvironment. You see on the far right is also a case of a human liver, where we had received a surgical resection from our colleagues at University of Washington of hepatocellular carcinoma, and also characterized. And you would see notice that there's a lot more heterogeneity in the human tumors compared to these cell line derived mouse tumors. But in all cases, we were able to see that the slices represent the similar sort of distribution of various cell types. Now, these cell types also change over time. So, for example, T cells will start to migrate out of slices in 3 to 5 days. You can actually start to collect them in the media, but a lot of them are still in the slices. NK cells start to die fairly quickly, about 3 to 5 days. macrophages and fibroblasts survive a very long time in slices. Now, that also depends on the various different cancer types and the tumor that you study. So as we are thinking that we are a pharmacology lab, we want to be able to use these slices for our pharmacological studies. So we had to also think about ways that we can assess the viability or assess the aggregate viability of slices. So for this, what we resorted to use is a reagent that is commercially available from Promega called real-time glow. It is a mixture of an enzyme and substrate complex, where enzyme is a cell impermeable and the substrate is a pro-substrate, which when enters the cell gets cleaved if the cells are metabolically active. And once it's cleaved, it comes out and reacts with the enzyme and it generates this white light or luminescence. So you can see in these images or the control treated slide or the DMSO treated slides, after 4 days, you can see a very strong signal of luminescence indicating high viability. And on the right next to it, you can see image of a slice that is treated with staurosporine. Now, staurosporine is a cytotoxic compound that inhibits more than 90% of the kinome. So after 4 days of treatment with staurosporine, a low concentration of 500 nanomolar, you can still physically see the architecture of the slice, but you see no viability signal coming from it. And on the right is a bar plot that shows a response to staurosporine in slices made from 6 different tumors, 3 from the NSG background, so immune compromised background, and 3 from bulb-C, in this case, would be an immune competent background. So showing the consistent responses to staurosporine coming from slices made from 6 different tumors. Again, showing consistency in response as a method to be able to assess the aggregate viability. Now, this method does not distinguish between killing or a viability of is it a cancer cell specific viability or is it some other cell type in the tumor microenvironment. So it is an aggregate viability assay, which is quantitative and robust. As shown here, one nice feature about this assay of looking at viability, that it does not end up lysing the tissue slice. So which means you can add this real-time glow the day you make slices or the next day, and then use a real-time assay to measure how the viability changes over time. And at the end of the assay, typically 3 to 7 days, you can take those slices and then use them for any downstream application. So for example, on the bottom right, the bar plot that shows the slices that were treated with the column PDX slices that were treated with staurosporine were then lysed and subjected to a protein array profiling by reverse phase protein array and looking at various different phosphorylation of various different signaling proteins, showing that the staurosporine treated slices, you see a reduced activation of phospho-ERK, STAT3, PKC, AQT, S6, as well as an increased activation of cleavcaspase 3, gamma H2X, and cleavcaspase 6, indicating apoptosis in the slices. So this is just one example for how these slices could be used for all other downstream or various downstream applications. You can also fix the slices and you can do imaging, so anything, or you can also do single cell sequencing. So the nice thing about this is that it is a real time and it does not end up lysing the tissue or destroying the tissue. So the architecture is maintained and the tissue could be used for any downstream application subsequently. So now we wanted to use the system that we have developed. We generate slices from tumors, and we have a method for delivering small molecules into the tumors, into these tumor slices, and we have a method to assess the aggregate viability. So I wanted to ask this question, could we use this model system to identify key interactions or key signaling proteins or key cell-to-cell interactions that are important in the slice environment? So the way we set up is we would do a drug screen in 2D culture cells, actually mouse cell lines, and then we would take these cell lines, inject them in mice, and develop tumors. And the idea is to be able to do a counter screen of the same drug library in tumor slices. And we're hoping that we would identify drugs that would be effective in tumor slices, but not in cancer cells. And we would use those drugs as a tool to uncover what drugs are, what pathways are they targeting, what cell types are they targeting, hoping to learn something more about that complex microenvironment that is important for the slice viability, but may not be as critical in the 2D culture environment. Now, one of the challenges of the slices, so when you're working with slices, is the amount of material that you get either from mouse tumor or from a human tumor is highly variable, and both in terms of its amount and also in terms of quality as well. So the large tumors tend to be very necrotic, so you cannot get a viable tissue slices from large tumors. So it is really a limiting factor of how many slices you're able to generate from any given tumor. So which is the reason they're not amenable for high throughput screening. You could probably screen in a slices made from human tumor, maybe half a dozen drugs to maybe 10 or 20 drugs, but it would be impossible to screen hundreds or thousands of drugs in the tissue slices of such scale. So this is to address this challenge, we combined a method that we had previously developed in our lab that exploits the polypharmacology of kinase inhibitors. I know this webinar is about tissue slices, so very briefly I will talk about how the approach works, then I will come back to how we are applying these machine learning based methods and integrating it with the screening in tissue slices. So polypharmacology is a term that broadly means a drug has multiple targets. So in an ideal world, if you have a drug that has a target, a single target, the target may affect a phenotype. The phenotype could be the viability of a slice. But in real world, what happens is that drug also has several other targets. And some of these other targets, or you can say off targets, can also affect the phenotype, such as viability of slice. So what we have in this approach, what we do is we take a series of drugs which have where we know their target profile, and we assess the effect of these drugs on a certain phenotype, for example, viability of slice. And then using machine learning approaches, we are able to then deconvolute what are the targets that contribute to the phenotype and what are the off-target effects. And for this, we rely on kinase inhibitors. Now, kinase inhibitors are, kinases are important enzymes that play a role in many biological processes and many signaling pathways. And when it comes to cancer, they're also enzymes that are frequently dysregulated in many cancers, either mutations or gene fusions or just sometimes expression. And more importantly, there are more than 90 kinase inhibitors that are now clinically approved. And over 50 kinase inhibitors are just recently approved in the past 5 or 6 years. So that gives us a lot of kinase inhibitors as a tool for drug repurposing and also for identifying roles of new kinases by exploiting this large set of clinically approved kinase inhibitors. So how this approach works is the first thing is we've done a large-scale profiling of a large number of kinases. So about 500 kinase inhibitors have been profiled against 400 kinases. So we know for each of the kinase inhibitors how well they inhibit around 400 kinases. Now, using this large-scale data set, we then computationally derive about 25 to 30 kinase inhibitors that best capture that space. So this immediately limit our experimentation from 500 kinase inhibitors down to 25. Because remember, I want to remind you that the idea here is that we want to limit our experimentation. Because as we're working with the complex models such as tissue slices, where we do not have sufficient amount of material to screen large number of drugs. So this is an approach that reduces your experimentation. So instead of having to screen 500 kinase inhibitors, we can screen just 25 inhibitors and use some machine learning approaches, such as elastic network regression and sometimes nonlinear modeling to help identify what are the key kinases and also predict the efficacy of kinase inhibitors that were not tested. So the whole idea here is to screen with a small number of kinase inhibitors and use the information of how these inhibitors affect various different kinases to build a model and to predict, to in silico predict kinase inhibitors that would be effective in any given phenotype. So this system or this approach can be applied to any phenotype. So in the past, just several years, we've applied a lot of this approach in some 2D culture phenotypes, such as proliferation and migration. And we've also applied this to macrophage polarization or change in cell state, or including EMT, also release of cytokines. So it's really very diverse approach that could be applied to any quantitative phenotype that you can measure in the presence of 25 or 30 kinase inhibitors. Now, for this webinar, I will be talking about our work that is currently under consideration about how we are applying this approach to assess a tissue viability, particularly tissue slice viability. So now I want to come back to our goal that I had previously defined. So the idea here was to generate tissue slices and use these tissue slices for a screen. And we would also do a counter screen in 2D cells. And our goal was to find drugs that would be differentially or selectively effective in tissue slices. And using these drugs as a tool to uncover some basic biology of cancer stroma interactions. And the whole reason we went into the polypharmacology approach is so that we could screen a small number of drugs and then use in silico models to predict the effect of large number of drugs. So we started with the 25 to 30 kinase inhibitors that were computationally designed to best to capture the space of the whole kinome. And shown here, the response of these 25 kinase inhibitor, showing controlled DMSO does not affect the viability of breast tumor slices. Erlotinib and EGFR inhibitor also do not affect, since this is a triple negative breast cancer model that is known to be not driven by EGFR signaling. And you can see statosporin, which is a multi-kinase inhibitor, completely kills the viability. And for this approach to work, you need to have these 25 to 30 kinase inhibitors that best capture the overall space. So if you have inhibitors that are all ineffective, you wouldn't be able to use this approach. Or if you have inhibitors that are all effective, so you need to cover the spectrum of both ineffective as well as effective, as well as somewhat effective drugs, so that we could use these in our model to then predict responses to other inhibitors that we have not tested. So using this, we build a model based on the training set, and then we use our model to predict responses to almost 500 kinase inhibitors. So shown here is a bar plot showing the model prediction of number of kinase inhibitors that would be effective, and how we define the effective kinase inhibitor, something that reduces the viability by at least 30% over 3 to 4 days of treatment at a low nanomolar concentration. What you will see is actually kind of surprising, that we found more number of drugs were effective in 3D slices than in 2D culture. For EO771, you see about 70 drugs that are more effective in 3D tissues, and just about 10 or 12 drugs that are effective in 2D cells. On average, this number was about 3 times more number of drugs are effective in reducing the viability of tissue slices than they are in 2D cells. Now, these are so this is out of 500. So you're only looking at about 10 to 12% of the drugs that are effective. So we do not see a broadly increase in efficacy of all drugs in 3D slices. So there seems to be some selectivity of which drugs are effective in 3D slices compared to 2D culture. So now had done this screen and Song Lee had done this screen with 3 different model systems. Two were a triple negative breast cancer model, so EO771, PY8.19, and one is a KPC model, which is a gold standard pancreas tumor model. And so you see there's a great deal of overlap between 2 triple negative breast cancer model and much less with the KPC pancreas model. But overall, we found 12 drugs out of 500 that were more effective in tumor slices and had no effect or very little effect in 2D culture. So what are these 12 drugs? Shown here in the bar plot. These are the 12 drugs that are more effective in 3D slices, as you can see with the box plots from all 3 models. If they are in the green, then they are more effective. If they are in the top part of the plot, they are less effective. So one of the drug, or some of the drugs were JAK inhibitors that we found, some were ROCK inhibitor, and some other inhibitors that found to be more effective in 3D slices at the same concentration and less effective in 2D culture. And one of the drugs that was found was consistently more effective in all 3 models in 3D slices was this drug called doramapimod. Now we have also expanded this to look at many other cell types, many other slices, a response of doramapimod in other tumors. So we had confirmed that it's not just a model prediction that doramapimod does decrease the viability of triple-negative breast cancer, KPC, as well as some PDX models, but it had a much less effect on some of these other less desmoplastic tumors, such as colon or MC38 or meningiomas. There seemed to be some specificity here that desmoplastic tumors were more sensitive to doramapimod. Now, as I mentioned earlier, doramapimod does not affect cell growth. So we had directly assessed this by looking at cancer cell growth in 2D and also in spheroid, and also looking at some large-scale data from the Broad Institute that doramapimod is not a drug that we would consider that inhibits the viability of cancer cells in culture conditions. So on the right is a bar plot that shows the response of doramapimod in almost 500 different cancer cell lines. Other than there's about 11 cell lines that show some response out of 500, and this is at 2.5 micromolar, which is 5 times higher than what we were testing in slices. Now, doramapimod is a p38 kinase inhibitor, and in our system, it does inhibit p38, so it does engage its target, but it has no effect on the growth of cancer cells, whether it's 2D or it's 3D. So then we set out to determine whether the effect that we're seeing in slices, so the reduced viability, does it also mean reduced tumor growth? So we did a series of experiments, and now we're trying to confirm the pharmacological response of reduced viability that we found in slices, whether we can validate them in vivo. Shown on the panel A is a response of doramapimod as a systemic high dose, where the mice were treated with 30 mg per kilogram over a 2-week period, and you see a slowdown of tumor growth. And on the right is a KPC model where we inject KPC cells directly into pancreas, and over a 2-week period, we look at how well doramapimod affect the growth of KPC tumors in pancreas. In both cases, we were able to see a decreased tumor growth in triple-negative breast cancer model as well as in pancreas model. And the bottom, you will see we also tested as a localized delivery of doramapimod. So doramapimod is a compound that was previously tested in the clinic for rheumatoid arthritis and some other immune disorders, and it targets p38. So we reasoned that it could have sort of more inflammatory response. So we wanted to directly assess what if we were to inject doramapimod directly into the tumor. And this time we injected very low concentration, or 1 mg per kilogram, directly into the tumor, and 2 different models. In 41 is a mouse triple-negative breast cancer, as well as a PDX, is a patient-derived xenograft model. At this very low concentration, it was able to still reduce the tumor growth. Now, these effects are not very large. These effects are actually comparable to the reduction in viability that we see in tissue slices. So we see about twofold. That's the maximum sort of decrease in the tumor growth in both of these models. So now we wanted to understand, so doramapimod is a compound that came from our screen that reduces the viability of tissue slices, reduces the tumor growth, but has no effect on cancer cells. So does it affect other cell types in the microenvironment? Or does it affect certain pathways that are only relevant in 3D microenvironment, but not in 2D? So first thing we did was we took these tissue slices and we did RNA sequencing. So the slices that were treated with doramapimod, we did not see a large-scale changes in the transcriptome. We see roughly about 200 genes that are changed, about 100 that are upregulated, about 100 that are downregulated. And if you look at the pathway enrichment of genes that were downregulated, we see a lot of ECM components are downregulated. So again, pointing towards, which was also confirmed by the ECM proteomics, or by both transcription level as well as proteomics, we were able to confirm that the ECM are reduced, the amount of ECM is reduced, and the genes that play a role in ECM regulation are also downregulated in response to treatment with dramapinode. Now, ECM is an important component in the tumor microenvironment. It provides a structural support. It also helps tumor grow, retain growth factors, and also provide a barrier for many drugs. And most tumors, especially solid tumors, have a very modified or different form of ECM that helps the tumor grow and invade the surrounding tissue. And one of the major cell types that is in the tumor microenvironment that contributes to ECM are known as cancer-associated fibroblasts, or CAFs. Then we asked, do the CAF fraction change with dramapinode? And indeed, we found that when we treat tumors or tissue slices with dramapinode, we see the CAF fraction reduce in these tumors. And then we started doing experiments directly in CAFs, which were derived from breast cancer and also pancreas cancer. And if you treat CAFs with dramapinode, we see much larger scale changes in the transcriptome, again, pointing towards changes in ECM, TGF beta pathway, and ECM receptor regulation, now suggesting that dramapinode is affecting ECM and likely affecting the CAF population, which is relevant and important in the slice culture environment. But as we grow cancer cells in 2D with a 10% DMSO, the CAFs are irrelevant and not important or not contributing towards the cancer cell growth. This suggests that using this complex model, such as tissue slices, is important both from physiological relevance and also uncovering important cell-to-cell interactions. Then we wanted to ask, okay, what is dramapinode targeting in CAFs? I also want to mention that as we do these experiments in CAFs, dramapinode does not kill CAFs. So even up to 10 micromolar, the viability of CAFs remains intact, but the amount of ECM that they secrete is drastically reduced. So dramapinode is a p38 inhibitor, as I mentioned earlier. It's designed as a very potent p38 family inhibitor, and our profiling, where we looked at how dramapinode affects over 400 different kinases. Indeed, we found that it does inhibit MAPK9, MAPK12, MAPK14. These are all isoforms of different p38 MAPK. And surprisingly, we found several other kinases that were as potently, if not more potently, inhibited by dramapinode. These included DDR1 and 2 and EphA7. DDR1 and 2 are the receptor tyrosine kinases that play an important role in binding to collagen or extracellular matrix. And when we knock down all of these targets to dramapinode and try to assess the function of CAF by measuring ECTA2, which is smooth muscle actin, or CXCL12, which is a secreted factor that is known to be secreted from CAFs. And what we found that it's the knockdown of DDR1 and 2 that decrease ECTA2, and also CXCL12, and also one of the p38 isoform gamma, which is a MAPK12. Knocking down of the other p38 isoform did not affect these things. And this was also confirmed when we knock down or overexpress DDR1 and DDR2 or MAPK12 in CAFs, we see an increased expression of ECTA2. Then we looked at the functional relevance of these kinases in CAFs. So we did these experiments now in a co-culture model, where we would take CAFs and we'll do knockdown of these kinases. So we knock down of all of these MAPK targets of dramapinode, as well as DDR1 and DDR2. And we would then wait for 2 days and then overlay fluorescently labeled, GFP labeled cancer cells on top. And then we would switch to a serum-free growth. What we find that is the 41 cells, without any serum, they do not grow, they start to die. But if they are growing on a layer of fibroblasts, they will continue to grow for several days. And if these fibroblasts had depleted levels of various different kinases, that could help support or reduce their growth. And we found that if we knock down DDR1 and DDR2 and MAPK12, specifically these three kinases, the CAFs were not able to support the cancer cell growth. Again, suggesting that these are as we have shown, these are the direct targets of dramapinode, and these kinases are important in CAFs in supporting the cancer cell growth. And we had some more subsequent work to show that trying to understand how DDR1, DDR2, and MAPK12 affects CAV function. Again, when we knock down these kinases in CAVs, we see a decrease in ECM, a decrease in the genes, expression of genes that play a role in ECM regulation. And if we were to overexpress DDR1, DDR2 in normal fibroblasts, we see an increased phosphorylation of MAPK12. So again, suggesting that all three kinases interact, and especially MAPK12 is downstream of DDR1, DDR2. It started to appear a sort of a linear pathway now that started from our work in tissue slices and went to CAFs and into a specific signaling pathway in CAFs that is important for ECM assembly and production. And it involves these kinases DDR1 and 2, which are the receptors for collagen, and a downstream effector, MAPK12. So it was unclear to us how do these kinases regulate ECM regulation. Then we started looking at what are the genes that are downregulated by all of these three kinases. And we found there were 12 genes that were sort of common, and these included several ECM components, some components in the immune regulation, and there was a one transcription factor called GLI1, which was downregulated when we knocked down either DDR1, DDR2, or MAPK12. And the GLI1 could also be upregulated when we overexpress these kinases. So suggesting that both knockdown and overexpression of these three kinases modulate the expression of GLI1. It seems to be a downstream effector. So very briefly, so GLI1 is a central component in the hedgehog pathway, which is an important pathway in embryonic development, where the levels of GLI1 are tightly controlled by this complex of SUFU and a series of kinases. When the presence of a hedgehog ligand, which binds to this patch receptor, which then inactivates, this is a smoothen receptor, which inactivates this degradation complex, and GLI1 is able to migrate into the nucleus. Now, there are several inhibitors that have been developed to target hedgehog pathways, some that target smoothen receptors, and some that directly target GLI1. And these are the ones that we're using in our studies. So, and previous studies have shown that the hedgehog pathway and GLI1 are important, especially in myofibroblast differentiation, in idiopathic pulmonary fibrosis, as well as in pancreas cancer, where a previous study has shown that fibroblasts, the hedgehog signaling is activated in these myofibroblastic or MyCAPs, where the ligands for hedgehogs secreted from epithelial cells or tumor cells and affect the CAF function and CAF, the ECM production in CAF. And indeed, what we found that when we overexpress this DDR1 and DDR2, we see not only increased phosphorylation of P38 that I had shown previously, but also a nuclear localization of GLI1. And in a similar co-culture assay, if you knock down GLI1 in calves, they're not able to support the cancer cell growth in 2D culture. Whereas if you knock down GLI1 in a cancer cell monoculture, it has no effect on growth of cancer cells. So now, with using our tissue slices that are a platform, using the pharmacological screening, we're able to identify an important role of calves in supporting the cancer cell growth. And using Dropinode as a tool that came from our screen, we're able to identify a signaling cascade that involves a DDR1-2 MAPK12, as well as a previously known GLI1 as a transcription factor, which plays a role in this canonical hedgehog pathway. But in this case, we found is also downstream of a series of kinases and play an important role in ECM assembly and production in calves, and also supporting the cancer cell growth. Now, one of the things I also mentioned earlier, that calves provide this barrier for tumors, for retaining for growth factors, as well as a barrier for various different drugs. And especially in desmoplastic tumors like pancreas cancer, it is known that calves could interfere in the access to targeted therapies, and include chemotherapy such as gemcitabine. We also then went back to our slice culture model and evaluated response to Dropinode in human tumor slices from pancreas patient. And shown here are the two plots where we're not looking at viability. Here we're looking at cleaved caspase 3 staining. So we're looking at apoptosis in tissue slices after 3 to 5 days of treatment with dronabinol. You can see in both of these cases, PDA47 and PDA49, we see changes either at a high level of dronabinol or 5 micromolar, or in combination with gemcitabine, where you see increased apoptosis in slices. And when we look at the same slices, if we look at the collagen staining using Mason trichome, we see that dronabinol treatment decreases the amount of ECM or collagen in these slices. Again, validating our molecular analysis that I showed earlier, that dronabinol treatment affects the CAP function and ECM production, also in human slices. And then we went on to do some studies to see if a combination of dronabinol and gemcitabine would be more effective, and indeed in reducing the tumor growth in a KPC, which is a gold standard model. Again, in this case, we injected KPC cells in mice, and after 1 week, the mice were enrolled in various different groups, either dronabinol alone or in combination with gemcitabine, where we were able to see an increase or we're able to see much stronger effect in reduction of tumor growth in the pancreas model when we combine with gemcitabine and dronabinol. So I hope I was able to actually convince that combining drug screening approaches in the tissue slices can enable a discovery of some basic understanding of cell-to-cell interaction. In this case, we'll focus on CAP and a specific pathway in CAP, and identify dronabinol as an inhibitor of a CAP function, and combining gemcitabine or chemotherapy with dronabinol enhances the tumor inhibition. Now, very briefly, just a couple of slides I just want to actually take back, because this is a webinar on tissue slices and organotypic tissue slices. So what I showed today is when we made slices directly from a primary tumor. Now, tissue slices also give us this opportunity to be able to generate slices from any organ size. So in our lab, we've been able to make slices from brain, lung, liver, etc. So which means that, it also gives us the opportunity to make slices not just from primary side, but also from metastatic sites. So I know in the cancer biology, we have cell lines that are derived from primary and metastatic sites. In a 2D culture, we already see a difference in these cell lines. But imagine what are some questions that you can begin to ask if you're working with slices made from various different organ sites. So if it's the same primary cancer cells injected in a colon versus injected in the liver versus injecting in the lung. So you have a tumor cells growing in a very different microenvironment that gives you this sort of physiologically relevant assay and model system to assess how tumor cells are interacting with the host site, or with the especially the metastatic site. Give you an example of a study that was published a few years ago that showed that the tumors or the cancer patient from melanoma or small cell lung cancer that had liver metastasis had a poor response to immunotherapy, whether it's PD-1 or PD-L1. And they went on to show that if they inject these MC38 cells subcutaneously in mice, they respond to a checkpoint therapy, such as PD-L1. But if they inject the same cells in the liver, they do not respond to immunotherapy. So the primary cell type is MC38, which is a workhorse model in oncology. If you inject them subcutaneously, they respond to immunotherapy. But if you inject them in the liver, so same cancer cells, but in a different site, they do not respond to immunotherapy. They went on to show that the microenvironment in subcutaneous tumor in the liver is very different. In the subcutaneous tumor, you have fewer macrophages and a lot more T cells that makes these tumor responsive to immunotherapy. And in the liver tumors, we have a lot more macrophages and fewer T cells. So we try to recapitulate this model system using tissue slices in our lab. From a postdoc in the lab, Yuling Huang went on to do these studies where we would take the MC38 cells, inject them subcutaneously, and also inject them either directly into the liver or through intrasplenic injection to generate liver tumors. So these are the same cells injected flank versus liver. Now we made slices from these tumors, and indeed we were able to see that the MC38 slices made from subcutaneous tumor did respond to PD-1, whereas the MC38 slices made from the liver tumor do not respond to PD-1 in a viability assay that I had described earlier. And shown on the right is also human colorectal liver that did not respond to immunotherapy, and showing SDS is a positive control star sporing that did respond even to slices made from liver. So just showing that the slices made from various different organ side, metastatic side, gives us the opportunity to be able to then probe the underlying biological mechanisms and to hopefully identify drugs that would be effective in inhibiting slice viability, slices that are made from the liver microenvironment compared to subcutaneous. So just to conclude this webinar, organotypic slices provides a physiologically relevant model system. It enables us to assess drug responses, and when combined with approaches such as the one that I described today, polypharmacology-based drug screening approach, it can enable screening of hundreds of or to thousands of different drugs, and also enable to discover new biology and help us understand cell-to-cell interaction, and potentially improve the rate of clinical successes. This, I would like to thank some members of the lab with the work I presented today, Song Lee, Now, Yuling, Ramya, and also many of my collaborators and funding sources. Thank you.
Thank you so much, Dr. Gujral. And I just wanted to open it up to our attendees. There are about 12 people here, and what I can see for our waiting room. Please feel free. We don't have any questions in the comments, but I just want to jump in for the AI and machine learning part. Can you comment a little bit on the accuracy you've experienced over time with that type of analyses?
Yes. So it is the machine learning part is a sort of iterative approach. So we start with about 25 drug tests. So the idea here is, if you had unlimited amount of material, for example, 2D culture, then you could just go ahead and test all 500 kinase inhibitors.
Yeah.
But the idea here is that we want to be able to limit our experimentation because we don't have enough material, we don't have enough slices, we have enough slices to test maybe 10, 20 drugs. So it is an iterative approach. So we first build a model using 20, 25 drugs, and then see how, and then use that model to predict unseen drugs. And then we go back and test the model's accuracy. A lot of times the accuracy ranges 70 to 90%, and we try to strive for at least 80% accuracy. And sometimes the accuracy is maybe 50 or 60%. So in those cases, we go back and test additional compounds, and then use those compounds and feed them back into the model to improve its accuracy.
Got it. Very nice. We don't have any other questions, so I would just want to send you a big thank you for your time. And Professor Gujral here is joining us from the West Coast, so it's very early for him. I hope you have a great rest of the day in great weather. Thank you again.
Thank you.